Hello everyone, good afternoon and welcome to JoyBeans, Conversations with Joy, or basically Conversations with me. Yesterday, I decided to do something different for my vlogs, and that is to do a series of short lectures on a number of basic concepts in genetics. So I'm handling two or three sections of genetics this semester, and I think that some of my students may find this lecture series useful. So I decided to call this lecture series Genetics in Small Bytes because we will be talking about, simple things that need to be very digestible and easily understandable. So this would be discussion on basic concepts in genetics that would come in small doses para chewable siya or madigest siya. And I'm hoping that every lecture will be under 10 minutes or at most around 12 minutes. And that is why this is genetics in small bites. And for the first lecture, the first topic is on parentals and recombinants in crosses. I found out nga na ay mga estudyante di ay nga medyo galibog pa unsaon pag-identify sa parentals and recombinants in crosses. And before I would proceed, I would like to be pointing out to you two things. In this lecture series, mag-try ko og explain ani sa Bisaya as much as I could para mas masabtan sa tanan. And for those who are not even my students or have no background in genetics, basig ma-appreciate pa pud nila kaning atong lecture series on genetics in small bites. And another thing is that I would like to point out to you that the photo nga nasa slide is actually a photo of a very common flower. I used to really love this flower when I was growing up. Abundant kay siya sa palibot sa unang panahon. This is the Mirabilis halapa, which is also called the 4 o'clock plant. It's a beautiful ornamental, and actually I have started using some of the fresh blossoms as vegetables last year. So, isa pa na siya butang. Anyway, that's just an additional information. So, the first topic, if I may repeat, would be on parentals and recombinants in crosses. Alright, so magsugod ta sa monohybrid cross. And our assumption is that the gene that we will be studying will follow Mendelian patterns of inheritance, which means nga the dominant allele would be completely dominant over the recessive allele. And so the dominant allele is usually written using a capital letter, no? Or that's the gene symbol. And the recessive allele would be written in small case or a small letter. And another thing is that we assume nga kada gene is independent of other genes, which means nga dili sila gaka-influensahan sa uban nga genes. Now, when we do a monohybrid cross, ato lang tan-awon og studihan ang usa ka gene. And in this case, you can see that I'm using a very generic gene symbol, which is the first letter of the English alphabet, A. But we know that for organisms, especially eukaryotes, in every cell that they have, kabaluta nga, they would have thousands and thousands of genes in their cells. And most of those genes are found inside the nucleus. So, another thing, okay, P stands for parental generation, okay? So, for our given example, we have a homozygous dominant A for the female parent, and we have homozygous recessive A for the male parent. And I hope that you already know how to derive gametes, so I'll go directly to the F1. The F1 is heterozygous for gene A, heterozygous dominant to be very exact. Now, the F1 is already the recombinant, okay, the recombinant genotype, because you can see that it is not exactly the same as either parent. So remember, the female parent is homozygous dominant for gene A, and the male parent is homozygous recessive for gene A. But the F1 is heterozygous dominant for gene A because it has inherited a dominant A allele from the female parent, and it also has inherited a recessive A allele from the male parent. Now, because I would be using plants as my examples, and so I would say nga ang F1, kung i-self-pollinate siya, it will, bear seeds, and then we would now be able to look into the F2. What would happen to this particular gene that has become heterozygous in the F1 generation or first filial generation? What will happen in the F2? Okay. So, kabalo na mo ani actually. The genotypic ratio in the F2 of a monohybrid cross would be 1:2:1, or ginaingon nato 1/4 dominant homozygous A, 1/2 heterozygous dominant A, and 1/4 homozygous recessive A. That is the genotypic ratio. Pero dili man guna nato makita. We just assume that that is true based on what we have learned from Mendel. Okay, because of the other laws like the independent assortment, that's the second law of Mendel, and the first law, which is the law of segregation. So that is why we would expect the genotypic ratio for a gene that follows Mendelian pattern to be 1:2:1. Pero technically, ang atong makita sa F2 population, kung tanaw na nato, tagsa-tagsa nato examine ang usa ka ang plants in the population would be the 3:1 ratio. So makita nato actually nga 3/4 of the population would be showing as the dominant phenotype, and 1/4 of the population will be showing as the recessive phenotype. Okay? So kaning 3/4 sa population nga magpakita sa ato sa dominant phenotype is actually the sum of, 1/4 homozygous dominant and 1/2 heterozygous dominant. And I have highlighted already in yellow unsa diha ang recombinants. So sa F1, if I may repeat, makita na nato ang recombinant. Okay? And then sa F2, ang katunga sa population actually is recombinant. That is our assumption based on what we've learned. Pero di nato makita or ma-separate ang dominant na homozygous sa gene A ug ang heterozygous dominant sa gene A in the population kasi mix na siya. We have no way of knowing unless we go to the F3 or the third filial generation. Kana noon ma-separate na nato asa diha ang tinuod nga homozygous dominant para sa A o heterozygous dominant para sa A. But if we're going to answer the basic query on asa diha ang parentals o asa diha ang recombinants, very simple, no? In the F2 of a monohybrid cross, 1/4 of the population would be parentals, 1 would be like the female parent, and in this case, kay F2 naman ni siya, pareho sa lolo o lola. So, 1/4 pareho sa lola, 1/4 pareho sa lolo. And the recombinants in the F2 would be 1/2 of the total population. So, ang recombinant sa F2 is pareha sa F1. Exactly pareha sa F1. Heterozygote po siya. So, I hope klaro na sa inyo. From the monohybrid cross, let us go to the dihybrid cross. And this is the first dihybrid cross nga atong discussan in this lecture. Again, I'm using very generic gene symbols. And now we are going to be studying or looking at two genes A and B. And ladies first, in the parental generation, the female parent is homozygous dominant for genes A and B, and the male parent is homozygous recessive for genes A and B. And again, assuming that you know how to derive gametes, I'm showing you that the F1 is heterozygous dominant for genes A and B. Now, nganong ing-ana man na siya? Kasi 50-50 ang contribution sa kada parent. So, if we look at the F1, asa niya nakuha ang alleles nga dominant A and B, ug asa niya nakuha ang alleles recessive A and B. Okay? Kabaluta nga ang dominant alleles A and B nakuha sa F1 from the female parent, ug ang recessive alleles A and B nakuha niya sa male parent, sa iyang papa. Now, assuming that this is a plant and it could easily self-pollinate and self-fertilize, then we can immediately evaluate the F2 generation. But instead of showing you first the genotypic ratio, ato sang tan-awon ang phenotypic ratio. Ngano gali? Because, the phenotypic ratio would be the thing that we would actually see. The genotypic ratio is just theoretical. Although we know that the genotypic ratio would be reflected, technically speaking, as the phenotypic ratio. And you have been encouraged, to be very familiar with the ratios of monohybrid crosses, dihybrid crosses, trihybrid crosses, even tetrahybrid crosses. But for a dihybrid cross that follows Mendelian pattern of inheritance, the expected phenotypic ratio in the F2 is 9:3:3:1. Or to be very exact, 9/16, 3/16, 3/16, and 1/16. So, unsa man na sila nga ratios. Before I elaborate on the, component genotypes or phenotypes per ratio, ato sang i-recall nga sa F2 generation, we should be able to see 4 phenotypic classes, no? Upat ka grupo sa mga individuals or organisms nga magpakita sa ato og lahi-lahi nga mga sets of phenotypes. So, majority of the F2 population, 9/16, that's more than 50%, would show us the phenotypes for the dominant A and the dominant B genes. Okay? So, you already know what the bar means, no? A 9/16 is a combination of the homozygous dominant for A and B and heterozygous dominant for A and B. Okay? So, it means ang bar, if I'm just going to, elaborate a little bit, it means that dominant A, and then regardless of the pair, whether another dominant A or a recessive A, would show us the same phenotype, the dominant phenotype for A. And for the B, for the dominant B allele, regardless of the pair, whether another dominant B or a recessive B, would show us the dominant B phenotype. So, unsa man ang 9/16, dominant A and B? Technically, that is a parental. And specifically, it's a parental type na pare-pareho sa lola o pareho sa F1. Okay? Because remember that 9/16 is a mix. And then, asa ang isa ka parental? Now, 1/16 is exactly the same as the lolo, okay? Recessive A and B. But they are the least number of plants in the F2 population. 1/16 is around 6.25%. Okay. Now, what are the recombinants? Of course, you can see what are the recombinants because they're already identified. But the recombinants, from the term itself, mga phenotypes or mga organisms nga dili exactly liwat, no, sa parents or sa grandparents. All right. 3/16, which would show us the dominant A phenotype and the recessive B phenotype, is the first recombinant. Okay? Wala siya kapareho either in the first filial generation or in the parental generation. And then the other 3/16 would be plants that would show us the recessive A phenotype and the dominant B phenotype. And again, wala sila kapareho sa parental generation o sa F1 generation. And therefore, they are the recombinants. Okay. Okay, so we are still on dihybrid cross number 1 and medyo saba na ang palibot. Gatsaba na ang mga insects o daghan na kaayong mga vehicles galabay. And unfortunately, I don't have a soundproof room to record this lecture, pero sagdi lang padayon lang ta. What we're going to look at in this slide would be the genotypic ratio of the F2 generation sa dihybrid cross. And para sa inyo, especially sa mga students na ako, ato na ning gidiskussan, nga you can use either the Punnett square or the fork line method to derive the ratio and you will come up with exactly the same sum or total, no? And pwede mo maglitan niya kung familiar na kayo mo sa ratio or na-memorize na niyo ang ratios. Pwede na mo maglitan niya nga 1:2:1, 2:4:2, 1:2:1. But of course, kada number na associated nga genotype, no? Kada number. So dapat inyo po ng kahibawan unsa ang 1, unsa ang 2, unsa ang 1, okay? Nga mga genotypes and so on and so forth. Now, mangutana mo nga nung maghimo magitag ing-ani nga mga crosses. What is it for? Okay? In plant breeding and animal breeding and other forms of breeding for other organisms, the reason why, no, especially for agriculture, the reason why nga nung nga breeding ta is because we would like to develop better types of organisms, and in this case, plants, we would like a better plant type nga dili nato makita sa parentals. Now, for example, if a plant that has a dominant A gene is resistant to disease A, but having the dominant B makes it also prone to lodging. So that is a case of having a positive trait and a negative trait together in one plant. Now, that's the case for the female parent in the parental generation. It is resistant to disease A, but susceptible or prone to lodging. You don't want that, especially if you know nga there is a gene for less susceptibility to lodging, and we assume nga, kunyari, having the recessive B gene would make the plant less prone to lodging. So, what do we want when we make the cross? Okay, we would like to develop a new plant type nga homozygous dominant para sa A, but homozygous recessive para sa B nga gene, making the plant still resistant to disease A, but no longer prone to lodging. And so, we ask, can we find that already in the F2? The answer is yes. Okay, it's the first recombinant, basically. It's the third in the left column. But, technically, dili ni nato siya ma-separate sa uban nga mga tanom nga heterozygous dominant for A and homozygous recessive for B. Kinahanglan pa ta mo adto sa third generation para atong ma-identify or ma-separate ang homozygous dominant A and homozygous recessive for B gene from the heterozygote dominant for A. Okay? So, hopefully inyo nang nasabtan. But, going back to the original question, asa ang parentals, asa ang recombinants diya, dapat nasabtan na or dali na nato makitaan nga ang parentals, syempre, either pareho sa mga lolo ug lola or pareho sa F1. But the recombinants are those that are not like either parents in the parental generation or not even like the F1. I hope that's clear. Alright, so we come to dihybrid cross number 2. And right off the bat, you can see that I am using a different second gene. It is gene C. Because in dihybrid cross number 1, we use genes A and B there. Now, another thing to take note is that the female parent is homozygous dominant for A, but homozygous recessive for C. And the male parent is homozygous recessive for A, but homozygous dominant for C. Now, those are really different parental genotypes compared to the first dihybrid cross, because every parent here has a dominant gene and also has a recessive gene. Now, again, assuming that you know how to derive gametes, the F1 of this particular cross is heterozygous dominant for A and heterozygous dominant for C. And it's highlighted in yellow, and therefore it is clearly a recombinant. But what happened to the genotypic ratio? The ratio would always be the same. 1:2:1, 2:4:2, 1:2:1, although the genotypes would differ, but the proportions would be the same. Now, I haven't highlighted the parentals or the recombinants in the F2 genotypic ratio. But if you look at the phenotypic ratio, so we have again the same, 9:3:3:1. Alright, so what is the 9/16? What's that phenotypic class? That is the phenotypic class that would show us the dominant A phenotype and the dominant C phenotype, which means that it is similar to the F1, but it's not similar to the parental genotype. So that means that it is a recombinant. And then 1/16, having the recessives A and C together, is also a recombinant because it's not the same as any of the parentals in the parental generation. So where are the parentals? The parentals would be 3/16, okay? Very similar. The first 3/16 is having the dominant A phenotype and having the recessive C phenotype, which is similar to the female parent. And another 3/16 showing the recessive A phenotype and the dominant C phenotype, which is similar to the male parent or the lolo, and that is a parental. So, karon kung nasabtan ninyo unsa nang recombinant, unsa nang parental, tanaw naman gud ninyo, no? Dito ra gid mo mobase sa parental generation. And you can exactly, and not exactly, you can very accurately identify which are the parentals and which are the recombinants. Now, in dihybrid cross number 2, assuming that we have a target genotype, a target recombinant genotype, and in this case, we want the dominant homozygous A and dominant homozygous C, because A again, dominant A codes for resistance to disease A, and a dominant C gene codes for better taste of fruits of this particular plant. And so we ask, if we have a population of 2,520 plants in the F2, what would be the expected number of plants nga homozygous dominant A ug homozygous dominant C? Although kabaluta nga sa F2, gasagol pa diri ang homozygous dominants ug heterozygous dominants. Pero wala lang, gusto na nato ma-estimate ahead, no? So, unsaon man na siya. So, in the genotypic ratio, kabaluta nga ang ratio sa homozygous dominant A and B would be 1/16, and that is technically 6.25%. Now, we can actually use that value to multiply with the total number of plants. So, we have 0.0625 times 2,520 F2 plants, and we can estimate that there would be around 158 plants. So, mag-expecta nga around 158 plants dapat sila ang homozygous dominant A o homozygous dominant C. Pero, i-emphasize nato nga, dili pa na nato sila ma-separate gikan sa mga heterozygotes dominant A o heterozygotes dominant C. Kay mix pa man na sila sa F2 population. Alright, so padayon ta, no? Kung based lang sa traditional evaluation, as I have said earlier, medyo lisod pag yun, ma-separate nato ang target nato nga recombinant genotype, which is homozygous dominant A and C, from the other plants nga magpakita pud sa phenotype sa dominant A and C. And there are 3 groups of heterozygotes, basically. So the first would be heterozygote dominant for A and homozygous dominant for C. The next is homozygous dominant for A, but heterozygote dominant for C. And the third is just like the F1, both heterozygous dominant for A and C. But it would be easier actually if we do another technique, which is DNA profiling and molecular assisted selection. So, dali ron ta nato ma-identify katong 1/16 nga grupo sa population nga immediately being dominant A and C homozygotes. Okay, but for the meantime, ang ato makita technically, based on traditional evaluation, would be that 56.25 of the population, or 9/16, are plants that would show us the dominants A and C phenotypes. And that would be around 1,418 plants. And if we still do the traditional evaluation, we need to go to the F3 generation in order to separate our target recombinant, which is homozygous dominant for A and homozygous dominant for C. And I'm not sure, but I'm hoping nga nakatabang ni siya. Murag mas nitaas pa din ni siya sa akong target nga 12 minutes. But maybe I'll get better next time. And with that, I would say until the next bite for Genetics in Small Bites. Have a good day, everyone, and I hope this helps make you understand a little bit of genetics. Bye.